Good afternoon. I'm grateful to Dr. Foster and the OIUF for their kind invitation and for giving me the opportunity to share with you some of the new therapeutic strategies in development in Burchard retinochoroidopathy or other types of non-infectious uveitis. These are my disclosures. To begin with, autoimmune diseases are the result of an imbalanced immune tolerance network, and treatment of these non-infectious types of uveitis remains a challenging issue. There are, of course, emerging pharmacological options administered locally or systemically, which are currently under evaluation, and some of them were presented previously before lunch in different types of uveitis. But I will try also to end my presentation by a new strategy which may be proposed to the patients and which relies on cell therapy in this type of indications. What are the optimal goals of therapy? Of course, we want to propose the best treatment modality which will be adapted to each type of clinical situation in order to achieve long-term control and complete control of intraocular inflammation by limiting as much as possible the use of systemic corticosteroids when this is possible, or taper the dose to 5 milligrams per day of prednisone to limit side effects, and this is of course very important, and specifically when we deal with new options, new drugs, or new ways of administration or cell therapy. This would be a little bit like the Grail quest, and I don't know if we will succeed. We hope that one day we will be able to treat each type of uveitis, but it remains difficult. It remains difficult because we have a lot of limitations, and we need these prerequisites in order to improve our therapeutic management of the patients. We need an optimal knowledge of disease pathogenesis, which is still lacking in Burchard retinochoroidopathy. The pathological features in the patients are not very well known. As it was presented previously, there are just a few cases presented in the literature and present in the literature showing the importance of different types of lymphocyte infiltration in the eye of the patients. There are studies of animal models. The transgenic A29 mice was presented, other types of experimental models mimicking Burchard retinochoroidopathy. And something which is new and coming up more and more is the study of biomarkers in Burchard patients. This is just taken from Yvonne de Kozak review on the transgenic mice expressing HLA-A29, showing the importance of lesions at the level of the retina. And this is why, going back to the previous talks, we try to talk more on Burchard retinochoroidopathy than Burchard chorioretinopathy. This slide is given to me by Narsingh Rao. It's a slide on the retina of a patient where you see the infiltration with the different types of cells, especially lymphocytes, at the level of the retina, but also here at the level of the choroid with the granuloma you may see here or here close to the vessels. And this is not available on Medline. It's it was published with Steve Ryan in the Burchard chapter of the Retina book. The new formulation or administration routes for conventional immunosuppressors will be the topic I will start with. And the first one, of course, would be voclosporin, which was presented as the next generation of calcineurin inhibitor based on this publication in the Lancet in 2008 in patient with plaque psoriasis. And although this was positive and presented with 3 different landmark studies, one, the 01 in 218 patients with active posterior inflammation. The second one was in controlled patients under maintenance therapy, and the third one was proposed to patients with active anterior uveitis. The first one on active patients showed efficacy, significant efficacy compared with placebo, and this is just to show you the different doses of 0.2, 0.4, 0.6 milligram per kg compared with placebo, showing the mean change from baseline in vitreous haze, which was significant when we had the 0.4 and 0.6 milligram kg compared with placebo. However, the second study did not reach significance, and the FDA asked for a second set of RCT, which was a phase 3 study, 6 months, randomized, multicenter, double-masked controlled trial to evaluate the efficacy and safety of the drug, again in a group of active non-infectious uveitis involving the intermediate or posterior segments of the eye. This study included 155 patients in 56 sites across North America, Europe, and Brazil, and the primary endpoint was, again, change from baseline in vitreous haze at 12 weeks or at the time of treatment failure if earlier. And in this study, at the end of 2012, we got the results, and Luvenik did not separate from placebo, showing that taking this endpoint, it was not possible to show any difference with placebo. And we know that cyclosporine A was proposed for years in patients with birdshot retinal choroidopathy. So this introduced something which is very important to consider as we are trying to have randomized controlled trials in order to test the drugs and come up with results. The new generation of intravitreal fluocinolone acetonide implant is also coming up. This was shown by Hank Kaplan, Ozurdex, Retisert. The fluocinolone acetonide implant is now approved in diabetic retinopathy, and it has lower dosage. So it may decrease the side effect of high intraocular pressure and secondary glaucoma. It's more easily administered, and the studies are ongoing right now, so I don't have results to give you about that, but it may be something interesting to consider because the drug may last for 3 years, which is not the case of dexamethasone and Ozurdex. The intravitreal sirolimus, this is an mTOR inhibitor, which negativate also the modulation of VEGF transcription. It's, of course, important to consider because the drug is not really well tolerated when given systemically, and the intravitreal use may be proposed to the patient. This is the SAFE study, which is going on. This is the phase 3 trial. And we are expecting the results by early 2014. This is run by Santen. What about the next generation of biological agents? The biologics, for sure, and this was presented by Antoine previously, is a step forward compared with corticosteroids and conventional immunosuppressors. They are highly effective. They were proposed in the late 90s, changing the outcome of most autoimmune conditions such as B27 uveitis, Behçet disease, and juvenile idiopathic arthritis-associated uveitis. They will achieve controlled disease activity, but unfortunately, in most of the cases, they are not curative treatments. Interferon alpha, TNF blockers, and Behçet's uveitis are a very good example. And I will not go back to these two molecules because it was presented previously. But of course, you have many other cytokines you can consider: IL-2, IL-12, the IL-10 group, the IL-17 and the Th17 lineage, adhesion molecules, of course, and receptors. Daclizumab was first presented by our host, Professor Foster Group, 8 patients with birdshot retinal choroidopathy refractory or intolerant to conventional immunomodulators. And what was interesting is that the disease was stabilized or improved regarding visual acuity and resolution of vitreous haze in 7 out of 8 patients with a follow-up of 25 months. The immunomodulators were discontinued in 4 cases, and adverse events occurred in 2 cases, leading to drug discontinuation. Now, the drug is used more in multiple sclerosis right now and less in the indication of uveitis. There is another molecule which is very interesting in the pathogenesis of birdshot retinochoroidopathy, and this was published by the group of Aniki Rotova in the Netherlands on the intraocular levels of IL-17 and other pro-inflammatory cytokines in a group of patients with birdshot retinochoroidopathy. They analyzed 23 biomarkers in paired samples of the aqueous humor and the serum of patients, and they found elevated levels of IL-2, IL-17, IL-1 beta, IL-6, and TNF-alpha. And with this, a positive correlation between IL-17 and both IL-2 and IL-23. What I was telling you about the difficulty of going from the pathogenesis to therapeutic options is that we got recently the publication of the secukinumab essay trial showing that in 3 randomized controlled trials, the SHIELD one in Behçet's disease, 118 patients, ENSURE and ENDURE, the drug did not separate from placebo, especially with the SHIELD study, and this was the reason when the trials were stopped. So we know that IL-17 is important. We know that we can block IL-17, and we know that it's very difficult to perform the study showing that. I don't have the data specifically in the group of birdshot in ENSURE and ENDURE trials, but this will be interesting to consider. And I think that Novartis is trying to start again a study on anti-IL-17 drugs. Anti-IL-6, the rationale, of course, this is a biomarker of autoimmunity in patients with non-infectious uveitis. Tocilizumab, Actemra, is a humanized IgG1 monoclonal antibody. Efficacy in small case series, and a phase 2 RCT is planned with Actemra. This is the publication from ADAN in 5 patients with refractory cystoid macular edema, and 3 of them had the birdshot retinochoroidopathy. And what is interesting, if you go to macular edema in this patient with birdshot at baseline, you see that you need to wait long in order to obtain the effect, as at 1 month you have still no response. So you need at least 3 months and then 6 months to achieve control of cystoid macular edema. Gevokizumab, an anti-IL-1 beta monoclonal antibody, is also a candidate. It will be interesting to see what will happen in non-infectious uveitis patients. It's, of course, effective, and we know that in autoinflammatory conditions. And when we go back to the presentation of Henk Kaplan on the potential involvement of autoinflammatory mechanisms in the pathogenesis of birdshot retinochoroidopathy, this may be an interesting option to consider, because at the other part, it will also block Th17 activation. This is the iGuard program, which is going on now with, again, three different RCTs. iGuard A in active non-infectious uveitis, the iGuard B for Behçet's patients, and the iGuard C for controlled non-infectious uveitis. This is for iGuard A, the patient's profile. Again, we find this dangerous vitreous haze score superior to 2+, which we have to consider. 300 patients in more than 100 sites. And the primary endpoint is the proportion of patients with the 2-point reduction in vitreous haze score on day 56. And this is for iGuard C. We have 300 patients again, and the primary endpoint is the proportion of patients with occurrence of uveitic disease through day 168. what is interesting is that we have an interaction between interleukin-17 and also interleukin-21, 23 in patients with uveitis. And I wanted to cite this paper by Paul Young and Steve Foster, published very recently in the American Journal of Ophthalmology, showing that, and these are the analysis of different types of cytokine in the serum of the patients, and you will find IL-21, of course, at high levels in the patients, but also IL-23 and TGF-beta-1. So it will be given with the data I presented on IL-17, it will be something very interesting to consider regarding therapy of patients with birdshot retinochoroidopathy. And the anti-IL-23 is the ustekinumab, which is a human monoclonal antibody, efficient and safe in 2 RCTs. Again, we find here severe psoriasis. There is an increased risk of infection, but it's definitely something we have to consider. So what would be now the next step after these biologics? We have to propose probably a global approach, and we have to steer the immune system into a more tolerant path, highly specific and causally oriented, reducing the side effects, and this is something very important, lower therapeutic cost, which may be considered in some part of the world, and eventually try to cure the disease more than giving remission to our patients. So what would happen? Would we choose between corticosteroids, conventional immunosuppressors, biologics, tolerance therapy, or cell therapy? This is really the question for the next 10 years. What are the limitations of the previous strategies based on tolerance induction? Target pathogenic T cells reactive against only a single antigen or epitope. This is, of course, very difficult to consider in patients with uveitis because we don't know the antigen. There is no sufficient consideration of the apparent complexity and dynamics of the pathogenic autoimmunity associated with uveitis in our patients, multiplicity of the target antigens and the emerging epitope spread, and of course, the objective and the aim of all this tolerance induction is the induction of one of the cell types, which is becoming more and more important, and these are Tregs. Aim of tolerance induction, it's very difficult to perform that directly, so we have to focus on indirect induction of regulatory T cells, and these cells are playing a major role in the control of intraocular inflammation. This is just a list of all the cells and the T cells we are working with, and the Tregs are appearing at the top of the list now in order to induce tolerance in our patients. And I will finish this presentation by proposing these few slides on an indirect strategy for tolerance induction. We started in the early 2000s by developing a model of experimental autoimmune uveal retinitis in bulb C mice. We injected subretinally hemagglutinin with the preparation of AAV, and just 30 days later, we induced uveitis, experimental uveitis in these mice by activated hemagglutinin-specific effector T cells, and we analyzed the eyes after 2 weeks. What was interesting is that in almost all bulb C mice without any genetic background, we obtained intraocular inflammation, which was evaluated with a clinical score, but also histologically, as you may see here, compared with the non-injected, non-induced animal. What was interesting is that the endogenous depletion of Tregs controlled intraocular inflammation, as we did that 10 days before inducing uveitis. And in that case, we obtained a disease which occurred earlier and which was more severe. This was published in Journal of Immunology, and we tried to see if it would be possible to consider induction and use of Tregs in animals and then in patients with uveitis. The problem was that we were using first systemic Tregs, specific Tregs, but we don't, as I said, we don't know the antigen in humans. So we tried to see in different conditions if it was possible to induce remission or control of inflammation by using a preparation of polyclonal Tregs. And what was possible is to achieve control of inflammation when we used polyclonal Tregs activated and injected intravitreally into the eye of the mice. Showing you here the difference we may see with the polyclonal Tregs activated compared with the polyclonal Tregs non-activated and the control. So that was a breakthrough, and we tried to go further and to propose, based on this point on Tregs and also this publication from the immunology group of our hospital, in the use of very small amounts of interleukin-2 in patients with HCV-induced vasculitis, and by increasing the systemic level of Tregs, they were able to control the disease, and this was published in New England Journal of Medicine. Then we proposed a clinical trial, this UV-REG, phase 1-2 study. The main outcome measure is, of course, tolerance at this time. We are including patients with non-infectious uveitis with a major visual loss. We discontinue immunosuppressors about 3 weeks before injection, and the level of corticosteroids is below 10 milligrams per day. We perform a lymphopoiesis at day -2, 2 days before injection, and then the lymphocytes are selected in order to obtain the Tregs. We activate overnight, and there is an intravitreal injection at day 0 with clinical monitoring and AC tap for cytokine analysis. The design of the study is 3 cell concentrations and 3 patients per group with an increasing manner, 0.4, 1.2, 3.6, 10 to the 6 cells, depending on the tolerance, and we start every time with 3 patients in each group, and we have to finish one before going to the next step. And we have 9 to 18 patients to include in this tolerance study. The outcome measures are the SUN criteria, the visual acuity, laser flare photometry, SDOCT for macular alteration, fluorescein angiography, and of course, biomarkers. The preclinical runs have been performed in 7 patients to validate the procedure of Treg purification with a yield of 86 to 90% CD4 plus, CD25 plus FOXP3 regulatory T cells, and to show the microbiological security of the process. The regulatory issues are now over because we obtained the authorization from the French agency. The first inclusion was in April 2013, with now 3 patients injected, promising results regarding safety, but we are just starting, and we need to look at the 3 cell concentrations before going forward. To summarize, I would say that all new alternatives lay the foundation for the next generation of therapies beyond the currently used biologics. The objectives are complete cure with safety, design tailored therapeutic strategies based on analysis of relevant biomarkers. And I think that this is really something very important. In order to go further, we need to know which type of uveitis is driven by which type of cytokine or which group of cytokine, in order to propose to those patients the specific molecule or the combination of molecules which will control inflammation eventually. So we have also to think of reestablishment of tolerance in autoimmune uveitis, which should be the final and realistic target. With this, I would like to thank all my coworkers and those who worked on the level of the lab with all we performed and developed since 2000, and also at the clinic. Thank you very much.